Welcome to lecture topic 2, methods for studying microorganisms. Again, if you haven't had the opportunity, you can download and print the notes, a copy of all of these slides if you'd like to take notes on during this lecture. All right, methods for studying microorganisms. A couple of terms that you should be very familiar with. The first here, culturing. It's not on the slide, but it's a term that we use all the time. Culturing. When you're culturing, you are working with microbes in the lab. It's a non-specific term. Basically, everything we talk about today will be under the description of culturing or working with microbes in the lab. Inoculation. To inoculate something. When you inoculate something, you introduce a tiny sample into a container of medium so they can reproduce and grow. So you're putting a small amount into a new container of media so that organism can grow in that container. That's inoculating or inoculation. Now you could inoculate a solid surface such as a petri dish that you see here. You can inoculate a liquid such as a bottle of blood or liquid media, or you can inoculate an animal or a bird embryo too. So it's introducing that small sample so that it can grow in that environment. The sample that you're introducing could also be called the inoculum. Once you have inoculated that media, the next thing we generally do is incubate it. Incubation is when you place something in an environment that encourages growth. So incubation is placing it in an environment that encourages growth. Very commonly when we talk about incubation, we're thinking of temperature. In general, warmer temperatures encourage growth while cooler temperatures discourage growth. Again, generally warmer temperatures encourage growth while cooler temperatures discourage growth. But it's not just temperature when we think about incubation. Incubation can also mean putting it in another environment that increases the growth rate. So it could be the right gas concentration. Many microbes require oxygen just like you and I require oxygen. And so placing them in an environment with the right oxygen concentration would be encouraging growth or incubating. You could place it in an environment without any oxygen if it was an anaerobe where oxygen was poisonous to it, or in the right amount of humidity. These are all things that would qualify as incubation too. Incubation just means putting it in an environment that encourages growth of that organism. Another term that we use all the time, or really an idea that we use all the time in microbiology, is the idea of isolation. Now, of course, we're working with very small organisms, too small to be seen with the naked eye. And generally speaking, organisms don't live on their own. They don't live one by one out in the environment. They're actually living in populations that are mixed with multiple different kinds of organisms together. In other words, if you looked at this group of microbes, you would see several different kinds. You'd see the yellow rod-shaped ones, and you'd see the white round-shaped organisms all together. Well, it's very difficult in a laboratory setting to study an organism if they're all mixed together. You can't tell who's doing what. You can't tell which one's causing your test result. So very often what we want to do is isolate them. We want to separate them out so that we can study one microbe at a time. The key to isolation is dilution. Dilute, dilute, dilute. What we want to do is take this sample that has multiple organisms in it. We want to dilute it so much that we separate out individual white round cells and individual yellow rod-shaped cells. So we want to dilute, dilute, dilute. And when you dilute it to the point that you have individual cells, then you place those cells on the culture of on the surface of your media. You inoculate them onto the media, put that into the incubator, and in the incubator you're encouraging growth so that one rod-shaped organism becomes 2, and then it becomes 4, and then it becomes 8, and eventually you wind up with a whole big group of microbes. And eventually that group is so large that you can actually see it with the naked eye, and that forms what we call a colony. A colony is a macroscopic group of genetically identical cells that arose from a single cell. So colony is a macroscopic group of genetically identical cells that arose from a single cell. You can see individual colonies. Using this dilution technique in order to isolate, you can dilute your mixed sample until you get individual cells, drop those individual cells on the surface of the media, and in the incubator, each of those cells will form a colony. All right, that's a macroscopic, which means that you can see it with the naked eye, a macroscopic group of genetically identical cells. They're all identical to one another because they arose from one cell originally on the plate. Now, after you've gone through isolation and incubation, you're going to inspect or look at what you have, what grew. Many times different organisms have different kinds of colonies. They may be larger colonies or smaller colonies. They may have a distinctive color. They may be very perfectly round in their shape, or they could be irregularly shaped. These are things that help you to identify what organism you have and how many organisms you have on your plate. For example, this plate here, if you look at this plate, you can clearly see that there are two different organisms, at least two different organisms on the plate. Notice that there are organisms that form yellow, shiny colonies, and there are organisms that form kind of dull, cream-colored colonies. So definitely there's at least two organisms on that plate. We would call that plate an example of a mixed culture. A mixed culture is a container with two or more species on or in it. So a mixed culture contains two or more species. Commonly what we're after in microbiology is a pure culture. A pure culture contains only one species. Again, it's generally easier to study organisms if you have them by themselves on your plate or in your broth tube. And so generally we're trying to get pure culture through this isolation technique. Now let's imagine that you've gone through isolation, you've then incubated your organism, you're now inspecting your plate, and let's imagine that you're trying to obtain a pure culture of just this red organism in this plate here. Right. So you think you've got a pure culture, you put it on the plate, you incubate it, you get it out, and you're looking at the colonies, and very quickly you can identify, yes, I have this red organism, but I also have somebody I didn't mean to have, somebody I don't want. We could call him a contaminant. A contaminant is an unknown or unwanted microbe. Again, a contaminant is an unknown or unwanted microbe. If you don't know what it is, but it's obvious that something is there that you don't mean to have, that would be a contaminant. And sometimes you do know what it is, but you want it to be a pure culture. So the fact that there's somebody else there makes it a contaminant. So the question is, if you wanted a pure culture of just this red organism, and obviously right now you have a contaminated culture, what would you do then? How would you get a pure culture of just this red organism? Hopefully what you're thinking to yourself is, well, I would take one of these red colonies off this plate, I would dilute it until I got individual cells, put them on the surface of a new plate. And absolutely that's right. You would go back through the steps of isolation. That would be called subculturing. Subculturing is taking a single colony and putting it onto new media. That's a subculture. Take a single colony and put it onto new media. The last step here in identifying or techniques that we commonly use in the lab, identification. Often that's the end goal of any of these experiments is to identify the organism that you've got. Ways that we can identify that organism. We can identify that organism based on its microscopic morphology. In other words, what's the cell shape? What's the cell structure? What kind of staining properties does it have? There's various different staining techniques that you can use to identify some of these organisms. Sometimes the colony descriptions are helpful if it forms red colonies or yellow colonies or big colonies or small colonies. That would help you to identify it. Another way that we can identify organisms is according to their biochemical abilities and their metabolic functions. Individual cells look very similar to one another, but often how they're different is what kind of metabolism they do. So we may be looking at, can this organism digest fructose? Or does this organism use urea? These are different biochemical pathways that we're looking at and investigating to identify the organism. And of course, if you have lots of money to spare, if you're in that kind of a situation, then DNA analysis, of course, would always be a very good technique to identify your organism. Generally, that's not available for most labs. Most of the time, we identify organisms based on their biochemical functions, so DNA analysis isn't always available for most situations. What about nutrition? What kinds of nutrients do microbes need growing in the laboratory or anywhere else for that matter? We can split up microbial nutrition requirements into sort of two different groups, what we call macronutrients and micronutrients. Macronutrients are those nutrients required in large quantities by the cell. The cell's got to get a lot of these nutrients because they play principal roles in the cell's structure and metabolism. So those are the macronutrients, the things that the cell needs a lot of in order to survive. The micronutrients, sometimes called trace elements, are required in small amounts. They're not generally part of the main structure of the cell, but instead they're involved with an enzyme. Some enzyme needs this nutrient in order for the enzyme to function correctly. So generally that's what the micronutrients are about. They're involved in enzyme function or maintaining protein structure, and the cell only needs them in very, very small amounts. If we look at the chemical composition of the cytoplasm of a cell is about 70% water. That's true of bacterial cells, that's true of fungal cells, that's true of human cells. In fact, all cells are mostly water. It reminds me of an old Star Trek episode where the alien life form said that humans were ugly bags of mostly water. Certainly, the ugly part depends on your perspective, I suppose, but yes, we are bags of mostly water, and so are all cells on the planet, including microbial cells. Lots and lots of water inside of a cell. What else is there? Proteins. The main structure and functional element of any cell is proteins. Proteins make up the structure of the cell, they make up the enzymes and the function of the cell. Lots and lots of proteins inside of the cell. If we look at the cell's composition, 97% of the cell is composed of only 6 elements. So only 6 elements make up the vast majority of the cell, and those elements are carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. If you have never learned this list before, let me point out that if you take the first letter here, CHNOPS, it spells schnapps. Which will help you to remember that. This is true not only of microbial cells, but all cells are composed mainly of those 6 elements: carbon, hydrogen, nitrogen, oxygen, and phosphorus and sulfur. And of course, it would make sense if the cell is composed mostly of these 6 elements. These 6 elements are the macronutrients. These are the elements that the cell must obtain in large quantities for it to function correctly, and that makes sense. It's made mostly of that stuff, so therefore it's got to get a lot of that stuff. Everything else is a micronutrient that's only needed in trace amounts. Let's look at carbon for a moment first. There are 2 different ways for organisms to obtain carbon on this planet. A heterotroph is an organism that obtains carbon in an organic form made by other living organisms. In other words, they ingest or absorb organic forms of carbon. You are a heterotroph. I'm assuming you did not photosynthesize your dinner, but instead you ingested organic molecules. You ingested organic molecules in your hamburger or your tofu or whatever it was. You were ingesting organic molecules, and that's how you got your carbon. You are a heterotroph. The other possibility here would be an autotroph. An autotroph is an organism that uses carbon dioxide, which is an inorganic gas, as its carbon source. Photosynthesis takes advantage of autotrophy, where they're using carbon dioxide from the atmosphere in order to get their carbon to make carbon-containing molecules. Autotrophs are not dependent on other living things for their carbon. Instead, they simply pull it out of the air. Plants and algae would be great examples of autotrophs doing that form of photosynthesis. So you can describe all organisms on the planet as being either a heterotroph or an autotroph. You can also describe all organisms on the planet as being either a phototroph or a chemotroph. Those terms have to do with how that organism gets its energy. A phototroph is an organism that obtains its energy from light rays. So this is photosynthesis. It's using photons of light as its energy source. A chemotroph is an organism that obtains energy from chemical bonds. So they break down chemical bonds and take the energy that was stored in those bonds. Some chemotrophs use very simple inorganic molecules like hydrogen gas or hydrogen sulfide gas. Some chemotrophs use complex molecules that were made by other organisms. That's what we do. That very same hamburger or tofu that you ate for dinner, you break that down to get carbon from it. You also break that down to get energy from it. So you are a chemotroph, getting energy from those complex molecules as well. So putting those four terms together, how do you get your carbon? How do you get your energy? You can describe all organisms as being one of four different types: a photoautotroph, a chemoautotroph, a photoheterotroph, or a chemoheterotroph. And this comes down to where do they get their carbon from and where do they get their energy from. So a photoautotroph, for example, takes the photo portion indicates that the energy source is light. The autotroph portion indicates that it gets its carbon from carbon dioxide. So a great example of a photoautotroph would be a plant or an algae, those organisms that are doing photosynthesis, as we've talked about it before. Another lifestyle, less common, but it does happen, a chemoautotroph. A chemoautotroph, chemo means it gets its energy from chemicals, in this case very simple inorganic chemicals, and it gets its carbon from carbon dioxide. Now, you probably can't think of an example of a chemoautotroph right away because they are kind of few and far between. Some deep sea bacteria do this, chemoautotrophy. Some archaea do this as well. It's a relatively uncommon lifestyle. A photoheterotroph, also relatively uncommon. Photo indicates that they get their energy from the sunlight. Heterotroph means that they use organic sources of carbon, so they get it from other organisms. So a photoheterotroph, it's a relatively uncommon form of photosynthesis that some bacteria do. So some bacteria live this lifestyle where they're not actually taking carbon dioxide from the environment, but they're using organic molecules in their photosynthesis. A chemoheterotroph. A chemoheterotroph, chemo meaning that they're getting their energy source from chemicals, usually large organic chemicals and compounds. Heterotroph meaning they're getting their carbon from that same location, from organic molecules. Chemoheterotrophs are the vast majority of things that you would think of. Animals and many microbes are chemoheterotrophs. Fungi are chemoheterotrophs. It's a very common lifestyle to live. So we talked about carbon and energy and how organisms can obtain that. What about the other macronutrients? Remember SNOTs: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. The other required macronutrients, hydrogen is required in large amounts because it's used for maintaining pH of the cell and also for hydrogen bonding. The more we learn about hydrogen and hydrogen bonding, the more important it seems to be. In fact, life forms would not exist if it wasn't for hydrogen bonding and those interesting qualities that water has. So absolutely, hydrogen is essential in the cell to maintain pH and to allow hydrogen bonding. Nitrogen is a component we find in proteins and DNA and ATP, all vital macromolecules inside of the cell. So of course nitrogen is essential. Oxygen is used in metabolism. Many microbes do a similar form of metabolism to what we do, so they require oxygen as well. Oxygen is also part of the structure of carbohydrates and lipids and proteins, so it's got to be there to form these macromolecules. Phosphorus is also part of the structure of both DNA and ATP, again necessary for those macromolecules to exist inside of the cell. And sulfur is an important component of some proteins, not all, and it's also an important component of many vitamins. So without it, things won't work properly in the cell either. Again, these are the macronutrients, the things that the cell needs in very, very large quantities. There are also, of course, micronutrients. There is a huge list of micronutrients that every cell requires. I want to remind you that micronutrients are not generally part of the structure of the cell, but instead micronutrients are usually needed as cofactors for enzymes. Micronutrients are usually needed as cofactors for enzymes. So enzyme number 1 does some important job in the cell, but it only works right if it has a molecule of potassium added into it. It doesn't function without that potassium. Enzyme number 2 does another function in the cell, but it only works right if it has calcium attached to it. Now you don't need much potassium and you don't need much calcium, but you need a little bit if you want those enzymes to function correctly. Those are the micronutrients or the trace elements. This is a small list of some of the micronutrients that are used in the cell. Just as a couple of examples here, potassium is important for membrane function. The membrane doesn't function correctly without a little bit of potassium. Sodium is used to transport things across the membrane. Calcium is used as a messaging system. Iron is used as a molecular magnet. These are just examples of some of the reasons that you have to have all of these micronutrients. This is not an exhaustive list. We could add a whole bunch of other micronutrients to this list. Zinc, copper, cobalt, nickel, molybdenum, manganese, silica, etc., etc., etc. In fact, if you go to your vitamin bottle right now, turn your vitamin bottle around and look at all of the things that are on there, all the different vitamins, all the different minerals, all of that stuff are micronutrients. You don't need a lot of it, right? But you do need a little bit. And why do you need it? Because various enzymes in your body must have it in order to function correctly. That's why it's so essential. Now, another topic that becomes important, especially with microbes. Some microbes require what we call growth factors. Growth factors are organic compounds that cannot be synthesized by an organism, and therefore you must provide it as a nutrient. Some organisms can't make everything that they need, so you have to provide that to the organism if you want it to grow. For example, maybe you're giving amino acids. Amino acids, again, are the building blocks of proteins. Some cells cannot make all 20 amino acids that are required to make proteins. Well, if they can't make their own amino acid, you better put it in the media if you want that organism to grow. That would be an example of a growth factor. Something extra, this organic compound that you added extra to this media. A vitamin would also be an example of a growth factor that you may add extra into the media in order to help that organism grow. So these organisms would be called fastidious. A fastidious organism, or tough to grow, is another way to say fastidious. So some organisms are fastidious or difficult to grow, and they require growth factors present in their environment if you want them to grow. So you got to add something extra in order to help those fastidious organisms grow. What kinds of stuff would you be adding? What kind of growth factors would you add for a fastidious organism? You might add blood. You might add a vitamin mix. These are things that contain lots of different nutrients in it to help out those fastidious organisms. You may add, I said vitamin mix, you may add an amino acid mix or extra vitamins. These are things, these are growth factors that you're adding to help out those fastidious organisms. So media. providing these nutrients in a laboratory setting, providing everything that's needed by that organism in order to grow in the lab. The most common media that we use is called nutrient broth. Nutrient broth is a liquid medium that contains beef extract. Beef extract is exactly what it sounds like. It's more or less beef broth, just like in cooking. It's beef extract plus peptone. Peptone is a vitamin and protein mix that's made from various different sources. So nutrient broth, liquid media containing beef extract and peptone. It's a rich environment. Lots of things can grow in it. Easy to grow stuff in nutrient broth. Nutrient agar. Nutrient agar is a solid media that contains beef extract, peptone, and one other object, agar. It's basically nutrient broth plus agar. Let's talk about what is agar. Agar is a complex polysaccharide that's isolated from red algae. Now, complex polysaccharide, I'm talking about a sugar, but it's a very complex sugar structure, not easily broken down. So agar is this complex polysaccharide that they get from red algae. We use it as a solidifying agent in nutrient medias. We can use that agar to solidify them. The value of agar. Why do we use it so commonly? First off, agar is solid at room temperature, so it allows you to make those petri dishes like the one you've inoculated in lab already. So it's solid at room temperature, but agar liquefies when you boil it. If you heat it all the way up to 100 degrees Celsius at boiling, it will liquefy, which is handy. It means that you can boil it, and then you can pour it into your petri dish, and then it will solidify in that dish, or whatever other thing you happen to want it to solidify in. And actually, it does not re-solidify until it cools to 42 degrees Celsius, which is very handy. We're going to do this experiment in the lab, but just briefly, if you add your microbes in to that liquid agar, right, if you boil it so it's at 100 degrees and it's liquid, if you begin to cool it down, right around 50 degrees or so, it's safe for you to add your microbes. It's not so hot that it will kill them, but it's still liquid. That means that you could pour your liquid agar containing microbes into a dish, and you could actually have your microbes growing in the agar if you wanted to. That's a handy thing. You'll see more about that in lab during week 2. We're going to start that process. Other valuable things about agar. Agar also provides a framework to hold moisture and nutrients. Carbohydrates retain moisture really, really well, so it creates kind of a moist environment for these organisms and for all of the nutrients that are in your media, so that's handy. The other thing that's really useful about agar is that it is not digestible for most microbes. Most microbes cannot digest agar, that polysaccharide. Now, why is that so useful? Imagine if you had your petri dish, your agar plate, you inoculated that agar plate, you put the plate into the incubator for incubation, and while it was in there, your microbes digested that agar. Well, what would happen is when you went to pull the plate out, you would have liquid because all of your agar had been digested. We don't want that to happen. So one of the really useful things about agar is that microbes don't digest it, or at least most microbes don't digest it. So that means that you can actually grow organisms on a solid media. That's actually one of the reasons we don't use gelatin. Gelatin is digestible by many microbes, and so therefore it doesn't work as well as agar. You can use gelatin, but it's not as handy because many microbes digest the gelatin. All right, some types of media. There are 4 main types of media here. A media could be called what we call synthetic versus complex. The term synthetic means that it contains pure organic and inorganic compounds in an exact chemical formula. In other words, in a synthetic media, you could tell me exactly how many molecules of carbon are in that media. You could tell me exactly how many molecules of nitrogen are in there. You could count it perfectly. That's a synthetic media. Right? We know exactly what's in it. If you were to make that media recipe here at PCC, you would wind up with exactly the same media as if you were to make it at the Pasteur Institute in Paris or at a medical facility in Bombay. Wherever you made it, it would be exactly the same. That's a synthetic media. That's different than a complex media. A complex media contains at least one ingredient that is not chemically definable, like beef extract. Beef extract is not chemically definable. You can't tell me exactly how many molecules of nitrogen are in that beef extract. In fact, if you get one shipment of beef extract, you may find a slight difference between the next shipment of beef extract. If you make this recipe here at PCC, you may get slightly different components in your beef extract than if you were to make this recipe in Paris. Right? So beef extract is not chemically definable. That makes it a complex media, anything that contains something that's not chemically definable. A general purpose media. A general purpose media grows a broad range of microbes. It's usually complex. It's meant to grow lots of different kinds of things. Nutrient broth and nutrient agar are general purpose media. The broth that you inoculated with the first day of lab is a general purpose media. It's meant to grow a bunch of different kinds of things. An enriched media. An enriched media contains complex organic substances such as blood, serum, hemoglobin, or growth factors, and it's required by fastidious microbes. So if you've added in some kind of growth factor to help a fastidious organism grow, we would call it an enriched media. All right. Selective versus differential. Other terms that we use a lot in the lab with regards to media. A selective media. A selective media contains one or more agents that inhibits the growth of some microbes and encourages the growth of other microbes. In other words, your selective media has something in it that selects for just one kind of organism. Only certain microbes grow on selective media. It's selecting for certain ones. A differential media allows the growth of several types of microbes, but it displays differences between the different types. So in selective media, only one kind of organism grows. In differential media, everybody grows, but somehow they look different, so you can differentiate between them. Let's look at an example of how this works. Let's imagine on the left-hand side here is where we'll start. On the left-hand side, we have a mixed sample with several different kinds of microbes in that mixed sample. It's a mixed culture. If we were to put that mixed sample onto general purpose media, then we would see everybody grows, right? General purpose, everybody grows. If we were to put that mixed sample onto selective media, only certain microbes would grow. Only the one type of microbe would grow. So we could select for the organism we wanted. If we were to put that mixed sample instead onto differential media, everybody grows on differential media, but you can now differentiate between different types of microbes. Some turn red, some turn purple as they grow on that media, some stay white. That allows you to differentiate. So everybody grows, but you can somehow differentiate between those organisms. Now, I want to go through a little practice using these terms. Before we start this, I want you to jot down somewhere next to you. As we go through these different descriptions of media, I want you to practice identifying if this is a synthetic media, a complex media, is this an enriched media, is it selective, and is it differential? Now, we're going to go through several different examples here. Please do not memorize the certain type of media that I'm talking about. What I want you to be able to do is, if I give you a description of a media, you should be able to identify, is it synthetic? Is it complex? Is it enriched? Is it selective? Is it differential? I just want you to be able to use those terms appropriately. So we're going to go through and describe some of these medias, and then we're going to go together and identify them. All right, so first here at the top, this is a blood agar plate. Blood agar plates start out just like nutrient agar. There's beef extract, there's peptone and agar in that media. Then they add sheep's blood cells to the media. So far, what do we know here? So far, we know that this is a complex media, and in fact, it's an enriched media because the blood cells are added to it. Now, why do they do that to this media? They add the blood cells because as some organisms grow on the surface of the plate, they digest those blood cells, and you can see these clear halos forming around those organisms. Other organisms do not digest the blood cells, and so you don't see the clear halo forming around them. That allows you to differentiate between those organisms that do form the halo and those organisms that do not. So blood agar plates would be complex, enriched, and differential because there's the ability to differentiate between those that digest the blood and those that do not. On the bottom picture here, you're looking at chocolate agar. Before you get too excited about what's in chocolate agar, let me tell you it's not what you think. Chocolate agar is actually a very similar recipe to blood agar. It has peptone, it has beef extract, there's yeast extract added to it as well as lysed red blood cells. So they take red blood cells and they chop them up, they lyse them before they add them to chocolate agar. So far, what do we know here? We know that this is an enriched media, this is a complex media, and why do they add them lysed? They add them lysed because some organisms, some very fastidious organisms, need those lysed red blood cells to grow. Now notice, this agar, even though it has basically the same recipe, chocolate agar is no longer differential because you've already chopped up those blood cells before you added them in. So it's lost its differential capability. All right, some more examples here. On the top, this is mannitol salt agar. The recipe for mannitol salt agar includes ammonium sulfate, potassium phosphate, a sugar called mannitol, sodium chloride. All of these things would be chemically definable, so this is a synthetic media. There is a high salt concentration in mannitol salt agar. The salt is there because most organisms can't survive in high salt, and only Staphylococcus can. So the high salt in this media makes it selective. It's selecting for those organisms that can grow in high salt. Now, mannitol salt agar starts out as this pink color, but there is a pH indicator and a dye added to it. If that organism is capable of fermenting, it will turn the media yellow. So that means it's also differential. So mannitol salt agar is synthetic, and it is selective and differential. The salt in it makes it selective, and the pH indicator makes it differential. You can differentiate whether or not your organism turns the media yellow. MacConkey agar on the bottom here. MacConkey agar's recipe starts out with ammonium sulfate, potassium phosphate, sodium chloride, a dye called crystal violet, and a pH indicator, all chemically definable, so this is also a synthetic media. The crystal violet dye that's in it inhibits organisms that we call gram-positive from being able to grow. Therefore, it is selective. It's killing off those gram-positive organisms. It also has a pH indicator in it so that some organisms will turn red as they grow, some will turn yellow, and some will stay brown as they grow. So it is differential as well. MacConkey agar is both selective and differential. One more example here of media. This is called chrome agar. Chrome agar is commonly used in a medtech lab to help diagnose urinary tract infections. The recipe for chrome agar is actually hasn't been published. It's under a copyright, but it is definitely a synthetic media. We do know it's synthetic, but I don't know exactly what all's in it. In addition to that media, there's a whole bunch of different dyes that are added into the media. And those dyes cause different organisms to turn different colors as they grow on the media. So what happens is you have a patient that comes in with a urinary tract infection. You take a urine sample from that patient, you put that sample onto Chromauger, and you can tell based on the color of the colonies what organism is causing the infection in your patient. So E. coli forms that red color, Klebsiella dark blue, Proteus peach, Enterococcus that green color, Staph pseudophilus pink, and Staph aureus cream. So you can identify very quickly what kind of infection your patient has. So this is definitely an example of differential media, allowing you to differentiate between organisms. All right, the next topic that we're going to talk about in this lecture is microscopy. Microscopy is something that we discussed in lab. So in some of these things I'm going to kind of go over very quickly because we already talked about it in lab, and in fact you are taking a quiz on that next week, week number 2. So hopefully this is a little bit of a review for you on microscopy. Now microscopy is really about two different factors: magnification versus resolution. Magnification is the ability to make something appear larger by bending light waves. We'll go through how that works in just a moment. And then resolution is sort of the clarity of the image. Resolution is the ability to distinguish two adjacent objects as separate. So magnification and making something appear larger by bending light. Of course, your microscopes do that through a series of lens systems. We did this in the lab, where if you go from the light source at the bottom. All right, the next topic that we're going to talk about in this lecture is microscopy. Microscopy is something that we discussed in lab. So in some of these things I'm going to kind of go over very quickly because we already talked about it in lab, and in fact you are taking a quiz on that next week, week number 2. So hopefully this is a little bit of a review for you on microscopy. Now microscopy is really about two different factors: magnification versus resolution. Magnification is the ability to make something appear larger by bending light waves. We'll go through how that works in just a moment. And then resolution is sort of the clarity of the image. Resolution is the ability to distinguish two adjacent objects as separate. So magnification and making something appear larger by bending light. Of course, your microscopes do that through a series of lens systems. We did this in the lab, where if you go from the light source at the bottom, the light goes through several different objects. The diaphragm would be first here, missing in this diagram. Diaphragm, then it goes through the condenser lens that condenses that into a solid beam. The light then passes through the specimen, goes through one of several different objective lenses. The objective lenses magnify that image by differing amounts depending on which objective lens you have in place. The light then passes through another lens system, the ocular, on its way out, and then eventually into your eye. So that's magnification in a microscope. Resolution, remember resolution is clarity of the image, or the ability to distinguish two adjacent objects as separate. Resolution has to do with an equation that you see here. R is resolving power. Resolving power is the smallest distance required between 2 points to distinguish them as separate. In other words, how far apart do two objects need to be in order to resolve the image clearly? That's resolving power. Resolving power, the equation here is resolving power is 0.61 times the wavelength of light that's being used divided by what's called the numerical aperture of the lens. Numerical aperture. Numerical aperture is a quality of a lens. It has to do with how good the lens is at gathering light. The numerical aperture is not something that you would measure. It's not something that you would calculate. It's something that's given to you. When you purchase a lens, the manufacturer tells you the lens's numerical aperture. So that's a number you plug into the equation. It cannot be changed. What can be changed in order to determine resolving power or resolution is the wavelength of light that's being used. And the shorter the wavelength of light, the smaller your resolving power, which means two things can be even closer together and still you're able to resolve the image. So in general, shorter wavelengths of light give better resolution. Therefore, we're always wanting to use the shortest wavelength of light possible in order to get good resolution of an image. On the right-hand side of the slide, you see a diagram that is showing you a kind of schematic of what we just talked about. If you're using long wavelengths of light, then the light can't get into all the nooks and crannies of this flea, and you wind up with a blurry, unresolved image. If you use short wavelengths of light, then the light does get into all the little nooks and crannies of the flea, and you wind up with a resolved, clear image. Right? So shorter wavelength is better. Now, when we're using visible light as our light source, we're always limited by how short a wavelength you can get from a visible light source. Best you can get is using blue light. That has the shortest wavelength that you can possibly get. So when we're talking about light microscopy, we're always limited by this fact, the fact that visible light only has so short a wavelength. Different types of light microscopy. There are several different types of light microscopy, all using visible light as the light source. Bright field microscopy. Bright field microscopy is what we've been doing in the lab up until this point. Bright field microscopy. Bright field microscopy is what we've been doing in the lab up until this point. In bright field microscopy, you have a specimen on your slide, and the light comes up from underneath it. The light passes through the specimen, goes up to the objective lenses. Some of the light is absorbed by that specimen, so you wind up seeing a dark specimen on a white background. Right? Dark specimen on a light background. That's bright field microscopy, what we're used to seeing. The value of bright field microscopy is that you can see true color. You can see what color an organism is using bright field microscopy. Dark field. Dark field microscopy. Dark field microscopy is more or less the same. There's just one extra lens system added into that microscope. In dark field microscopy, they're using what's called a stop condenser. The stop condenser blocks direct light and only allows refracted light to be seen. So in other words, light that comes up from the bottom through your slide, if it directly passes straight into the scope, that's blocked by the stop condenser. Light that hits something, your specimen, and bounces off of that specimen, that's picked up by the scope. So you wind up seeing a light specimen on a dark background. That's dark field microscopy. Now in dark field microscopy, why would people be using this? Why do you want to see a light specimen on a dark background? Most often, dark field microscopy is used in motility studies. It's very difficult to watch light-colored things swimming around on a light background. It's much easier to see light-colored things swimming around on a dark background. So often, dark field microscopy is used for studying motility. Phase contrast. A phase contrast microscope, excuse me, a phase contrast microscope has the added capability where it magnifies subtle changes in light density. So as the light passes through the specimen, the areas that are denser in that specimen absorb more light. The areas that are less dense absorb less light, and the phase contrast scope picks up and magnifies those differences. So in the picture here of the phase contrast scope, notice that what you can see are intracellular structures, because some parts are darker than others. And that's the cool thing about phase contrast scopes. You're not seeing true color, but you can see intracellular structures using a phase contrast scope. Now remember, with any of these forms of light microscopy, we're still using visible light, and therefore the resolution limits of visible light microscopy. Another type of microscopy that's used a lot of times in a medical setting, this is fluorescence microscopy. In fluorescence microscopy, you use a UV radiation source in order to hit the specimen and cause it to glow. Here's how it works. You take a sample from a patient. Take this sample, in this case you're looking at a cheek swab. Take that cheek swab, put it onto a slide. You then add a dye. You add a dye that very specifically attaches to only one organism. And that dye, when it's hit with UV light, glows. It gives off visible light rays. So you can see here there's one type of microbe that's been coated with a red dye so that when you hit it with UV light, it glows red. Here's another microorganism that's been coated with a green dye, so when you hit it with UV light, it glows green. This is very useful in diagnosing infections. For example, if you want to test your patient for syphilis, patient comes in, you suspect that it's syphilis, you take a little sample, you put that sample onto a slide. You then use a dye that only attaches to syphilis. It can't attach to anything else. You wash off the excess, hit it with UV light, and you're looking for glowing. If it glows, then you know your patient has syphilis. If it doesn't glow, you know your patient doesn't have syphilis. Now you don't know what it is, but you can identify syphilis or not using this type of technique. And in fact, you can purchase these dyes that attach to only specific organisms. You can purchase those online. They have all sorts of different ones. One company I was looking at earlier has dyes for syphilis, herpes, influenza, and a whole handful of other organisms. Now, in all of these cases, we're constrained by the limits of resolution with light microscopy and how specific light microscopy can get based on the wavelength problem. There was a huge change in biology once they came up with electron microscopy. Now, it turns out if you get electrons moving very, very quickly, if you force them to move at an extremely fast rate, as they move, they begin to move in a wave-like pattern. So a beam of electrons traveling in a wave-like pattern is what's being used as the source here in electron microscopy. Instead of using light, you're using a beam of electrons. Now, electrons are high energy, and when they travel in a wave, they have an extremely short wavelength. In fact, they're about 100,000 times shorter than the wavelength of visible light. Now, we know resolution is a function of wavelength. So if your wavelength is really, really short, this is going to mean that your resolution is really, really good. It means that you can see details in very, very fine images. So in fact, electrons have a tremendous power to resolve minute structures, because resolving power is a function of wavelength. So with such an extremely short wavelength, you have lots and lots of resolution ability. Because you can resolve images so well, now you can magnify them, and you're going to see the image clearly. So with an electron microscope, you can get magnification anywhere from 5,000 up to about 5 million times magnification, which is really stunning. That was a huge benefit to microbiology and cellular biology, the ability to see very small things very clearly using electrons. let's compare how this works versus a light microscope. Now in this diagram, if you look at the left-hand side, they're showing you light microscopy, sort of the opposite orientation that we've looked at before, but everything that we've seen before is still there. You have your light source, condenser lens, specimen, objective, ocular lens, and then of course the light goes into your eye. An electron microscope works very similarly. You have your source of electrons at the top on the right-hand side there. The electron gun is sending out electrons. You need those electrons to travel at an extremely fast rate in order to get them in a wave-like pattern, so they got to accelerate very, very quickly. Condenser lenses for an electron scope are just magnets. Remember that electrons have a charge, so they can use magnets to condense that beam of electrons. The electrons then pass through the specimen, they pass through an objective lens, they pass through an ocular lens. You really do not want your eyeball at the other side of this beam of electrons, so there is a viewing screen. The electrons hit some panels, and then that information is sent to the viewing screen. So a very similar idea, even though it's electrons instead of light beams, it's a very similar idea, moving things very, very quickly to make a wave-like pattern and then using this lens system to magnify it. There are two different types of electron microscopes, what's called a transmission electron microscope, or TEM, and a scanning electron microscope, or SEM. Transmission electron microscopes and scanning electron microscopes work slightly different. They're both using this beam of electrons, but the images that you get are slightly different because of how they work. So a TEM first, transmission first. In a transmission electron microscope, what you do is you usually freeze your sample, take very thin slices of your sample using a laser, and then you lay it down and you let the electrons go through it. Right? So what you wind up seeing is this cross-section. You see a cross-section of your specimen, because the electrons are actually passing through this slice that you've created. That's a transmission electron micrograph. For the scanning here, in the scanning electron microscope, what they do is they coat the outside of the microbe with something, usually some kind of metal ions, and then they shoot their beam of electrons at it, and the electrons bounce off those metal ions, and they bounce in all different directions. Based on where they bounce to, you can go back and look at what the outside of your specimen would look like. So in a scanning electron micrograph, you're looking at the contours or the outside of your specimen. So here's an example of that. So I would expect if you were looking at an image, you could identify if it was a transmission electron micrograph or if it was a scanning electron micrograph, because you would know if you're looking at a cross-section of your sample or if you're looking at the contours of the outside of your sample. I also want to point out while we're here that electron microscopes do not ever show color. Electron micrographs are always in black and white. Any color that you see in these images has been added by researchers trying to illustrate particularly what they want you to see in that picture. So all electron micrographs are black and white. Any color is added later. Preparing the specimen to view it under the scope. Two different ways that you can prepare a specimen, either what's called a wet mount or a type of wet mount that's called a hanging drop. In this case, what you do is you have live cells, you have your slide, you put the liquid with your cells in it on top of the slide, you slap a cover slip on top of that, and you look under the scope. Right? The value of a wet mount, it's easy, it's fast. You can see things moving around because they're still alive in that wet mount. The problem with a wet mount is that it is temporary. If you run out of time in lab, you can't come back and look at it next week. Once it dries out, it's game over. That organism is dead. So that's a problem. Also, it's hard to see a lot of things in wet mounts. Many microbes are clear, and looking at a clear organism swimming around on a clear background is extremely difficult. So wet mounts can be difficult for that reason as well. The other option here is a fixed mount. In a fixed mount, you take a thin film of your specimen and you dry it onto the surface of your slide, and then we call it a smear. This smear can then be stained using all sorts of different dyes, and you can stain your specimen whatever color you want once you have it in this smear form on the surface of your slide. Value here, of course, you can stain it whatever color you want to see things more clearly. It is long lasting. Smears will last just fine for decades without any problems whatsoever. The detriment is, of course, they're dead. You're not going to see anybody swimming around in a smear. And sometimes things change as they die, so you're not seeing the cell as it would be alive when we're talking about a smear. All right. Staining. If you have that smear, what kind of staining could you do? You could either use a basic or an acidic dye. Basic dyes have a positive charge on them. Acidic dyes have a negative charge on them. It turns out that most bacteria have a negative charge in their cell wall. So bacteria have a negative charge in their cell wall. If you were to add a basic dye to your specimen, it's positively charged, your bacteria is negatively charged, the dye would stick to the bacteria, and you would wind up with this sort of scenario where you have colored bacteria on a clear background. That's called positive staining. Positive staining uses a basic dye, and the dye sticks to the bacteria instead of the background because the bacteria are negatively charged while the dye is positively charged. The opposite would be an acidic dye. If you use an acidic dye with a negative charge, the charge is actually repelled from the bacteria because the bacteria is negative, the dye is negative. So therefore, it does not stick to the bacteria, but it turns out that glass has a slightly positive charge. So the dye will stick to the glass very effectively. You wind up with what we call negative staining. Negative staining is when you have clear organisms on a dark background, and acidic dyes do negative staining. A couple of other terms here. A simple stain. A simple stain uses only one dye to stain the organism. A differential stain. A differential stain is a process that uses two different dyes, and it differentiates between cell types or cell parts. So a differential stain uses two different dyes to differentiate between cell types or cell parts. A special stain is meant to emphasize a certain part of the cell. So it's meant to emphasize a certain structure in or on the cell. Let's look at some examples of these different kinds of stains. Top left-hand corner here, this is a staining process where you use crystal violet. Notice that the bacteria are stained purple on a clear background. That is a positive staining that's done with a basic dye. Crystal violet is a basic dye. It's also, did I say simple stain? If I didn't, I'll say it again. It's a simple stain, just one dye, just the crystal violet, and the crystal violet is basic, doing positive staining. Here's another stain in the center here. This is called a Gram stain. In a Gram stain, you use two different dyes. You use crystal violet, which is dark purple, and you use safranin, which is this sort of pinkish red color that you see. Get my pointer to work here. This pinkish red color is caused by the safranin. So Gram staining uses two different dyes, so it is a differential stain. Some organisms turn purple in the Gram stain, some organisms turn pink in the Gram stain. So it's differential. Both the purple and the pink dye are doing positive staining because the bacteria are colored on a clear background, and they are both basic dyes. On the right hand side on the top here, this is a staining process using India ink. India ink is an acidic dye, and it does negative staining. Notice that the background is stained black and the organisms are clear on that dark background. So it is doing negative staining, and it's an acidic dye. It turns out that this one, in this particular example, I would also call this a special stain. Remember, special stains emphasize certain cell parts. This stain is meant to show what's called the capsule. The capsule is this thick sugary coat that's around the outside of cells, right? If you notice, these cells sort of look like a fried egg. What would be the yolk of the egg in the center is the cell. What would be the egg white around the outside of the cell would be this capsule, and this is a staining process meant to show that. So